Good morning, madam. My name is Mugilan. My matrix number is 23DBT20F2007. Today, I'm going to carry out a simulation on DNA extraction. So, in this experiment, we need to isolate some DNA from a human test subjects for uh, okay. Scientists isolate DNA for a variety of reasons. Why they uh, extract the DNA is for genetic testing, body identification, and analysis of forensic evidence. DNA extraction is typically the first step in a longer laboratory process. DNA extraction is an important part of that process because the DNA first needs to be purified away from the proteins and other cellular contaminants. We need cells because that where the DNA is. Inside almost every cell in our body is nucleus, and inside each nucleus is about 2 meters of DNA. So this is the chromosome. So this is the histone. This called the 2-meter length of DNA strand. So for the DNA extraction, we're going to collect the samples from the mouth, which is the cheek cells. The skin on the inside of our mouth loses thousands of cells every day. These cells are we are after. So let's get started to the DNA extraction. The steps we will follow to purify DNA from a cheek swab are shown below. The first step is collect the cheek cell. The second is burst cell open to release DNA. Then third process is separate DNA from proteins and debris. And fourth procedure is isolate the concentrated DNA. Okay, here are the materials that we going to use for the DNA extraction. The water, the microcentrifuge, the buccal swab, the micropipettes, and the sample tubes like lysosyl solution, concentrated salt solution. Now we're going to carry out the first procedure, which is the collect cheek cells. So here, next we will be placed the swab into a epigraph tube and look closely at the swab. Note that it is covered with hundred of tiny cheek cells. Inside each cheek cell is a nucleus, and inside nucleus is DNA. Now we're going to pour the lysis solution into the tube. Then this is the warm water bath. Place the tube into the water bath. Okay, the lysis solution that we just added contains two important ingredients, which is the detergent and an enzyme called proteins K. The detergent disrupts the cell membrane and nuclear envelope, causing the cells to burst open and release their DNA. And the DNA is still wrapped very tightly around the proteins called histones, and the proteinase K cuts apart the histones to free the DNA, so the DNA will be released. The cells have stayed in the warm water bath long enough for the DNA to be freed from the cells, and we have removed the swab from the tube and add some concentrated salt solution to the tube. So we could see that the red strands are the DNA, and the brown color is the salt, the protein and debris. The salt causes the protein and the other cellular debris to clump together. So place the tube into the microcentrifuge. Then, in order to balance the centrifuge, a tube containing water is placed opposite your tube. So click on the lid. So we could see that this is the DNA and the liquid, and the bottom was is the protein and debris. Inside the centrifuge, the tubes spin around at high speed, the heavy clumps of protein and cellular debris to sink to the bottom of the tube, while the strands of DNA remains distributed through the liquid. Use the micropipette to carefully remove the top liquid which contain DNA and place it into a clean tube. The protein and other cellular debris stay behind. Now we're going to move to the fourth procedure, which is the last one. Isolate the concentrated DNA. This is the isopropyl alcohol. So add some isopropyl alcohol to the tube. Now we could see that inverting the tube several times mixes the isopropyl alcohol into the DNA solution because the DNA is not soluble, does not stay dissolved in isopropyl alcohol. It comes out of solution. So we can now see that clumped DNA with our naked eye. So now we're going to put the tube into the microcentrifuge. So then the DNA sinks to the bottom of the tube. So once the liquid is removed and the DNA is allowed to dry, so we can dissolve it into the solution of our choice. So try our hand at the DNA extraction using common household items that we can find in the kitchen.